In exercise 2, you used wet mounts to look at living organisms. Most of these were pond water representatives, and you also looked at specimens you prepared yourselves with yeast and bacteria. The objective of this exercise is to classify these organisms that you viewed and to see where they fit in among other organisms in the natural world. As you observe these organisms, you also recognize their means of nutrition and their means of motility. So you're going to be filling out a table with the 6 microorganisms that you viewed in lab. Remember, those 6 microorganisms were in 6 different general groups. We'll get to that in a moment. This is a representation of the table you're going to be filling in. It has 6 columns for you to complete, and then you're going to add the organisms in down the left side. So you viewed 2 organisms in lab as a mixture, the Saccharomyces, which was yeast, and the Escherichia coli bacteria organisms that you observed at 400x magnification. You also observed quite a few specimens, and you are going to include the 6 organisms that you observed and identified in the lab in these boxes. Depending upon the resource that you use to identify the organisms, you may only know the genus, but you also may know the genus and the species. So put whatever information you know. Remember that genus and species are going to be italicized or underlined. You can see that Saccharomyces is a genus name, so it's italicized. Escherichia is also a genus name, italicized, and coli is a species name, and it is italicized. If you're handwriting this assignment, you should underline the names of the organism. After you write the 6 organisms in these boxes, you're then going to proceed filling in the rest of the boxes. First is going to be cell type. As you complete this assignment, remember back to what you did in lab and think about what you saw. Don't try to go to the internet to research this information. You're going to get very frustrated if you do. Follow this tutorial when you fill this in and ask questions if you have any. The first column is going to be cell type. There are only 2 cell types, prokaryotes or eukaryotes. Bacteria and cyanobacteria are prokaryotes, and everything else is eukaryotic. The next kingdom is the Whittaker. The next column is the Whittaker kingdom. There are 6 Whittaker kingdoms. There used to be 5. The first one is Animalia, or animals, plants, or plantae, Protista, and you learned in lecture that Protista is very much a catch-all kingdom. Quite a few organisms fit into that one. Fungi. Fungi is also known as Mycetae. And then the old Monera kingdom has been split into 2 different kingdoms in the Whittaker system, called Eubacteria, or sometimes just called bacteria, and Archaebacteria, or Archaea. Finally, the most overarching category is the domain. There are 3 domains: Eukarya, or Eukaryota, Eubacteria, also known as bacteria, and Archaea. Eubacteria and Archaea are prokaryotes. They're bacteria. Everything else is going to fit into the domain Eukarya. Continuing with the boxes, the next column is the general group. The general group is not something you're going to find online. This is pretty much something I made up, but I want you to figure out how these organisms are classified. You should only have 1 organism from each general group in your table. You may have more than 1, and you can use those as bonus. I'll explain that in a moment. So let's take a look at the bottom here. At the bottom we see Archaea and bacteria. These are both names of domains as well as kingdoms. You can see with the bacteria that there are different shapes. So a general group for a bacteria would be bacillus, or rod-shaped, or coccus. These are terms that you learned in the first lab. So think back to what Escherichia coli looked like and then classify it in the general group. Was it a rod-shaped bacterium? Was it a coccus or a spirillum-shaped bacterium? Looking at Protista, you can see that there are quite a few different general groups. There are ciliates, flagellates. Flagellates are also called mastigophorans. There are amoeboid organisms. There are green algae, Diatoms. You probably saw diatoms in lab. If you looked at the prepared slides, you may have seen dinoflagellates. Perhaps if we had an aquarium specimen, you saw red algae. So these would be the general groups that you would find in the Protista kingdom. Remember, microscopic algae are classified as protists, not plants. So in this lab, everything we looked at was microscopic. As a result, you're not going to list anything as plants in this lab. The fungi can be yeasts or molds. Remember that you looked at Saccharomyces, so Saccharomyces is classified in the general group of yeasts. And then Animalia. You may have seen some Animalia in the specimens that you looked at. You might have seen a nematode when you looked at the vinegar eels. You also may have seen some rotifers. Rotifers aren't shown on this branch, but rotifers were those lovely carpet scrubbers that you saw, and they are in Kingdom Animalia, and their general group is simply a rotifer. The means of motility comes next. Remember, not all organisms are motile, so if the organism is not motile, put it in the box if that's the case. The means of motility for prokaryotes include flagella, endoflagella, gas vacuoles, and gliding. The mechanism of gliding is not very well understood, especially in the cyanobacteria. Perhaps you saw oscillatoria. Oscillatoria moves by a gliding type of motion that's not well understood. Eukaryotes also can be motile. Their motility may include appendages, cilia, undulating membranes, pseudopods, muscle contractions, flagella, and mucilaginous secretions from a raphe. If you saw pinnate diatoms, the ones that are pointed on both ends, sort of like a cigar, these are going to move via these mucilaginous secretions. Pay attention that just because you didn't see the organism move doesn't mean that it's non-motile. It may have been dead. Finally, you're going to determine the means of nutrition. Prokaryotes can get their nutrition by absorption or photosynthesis. Eukaryotic nutrition is by photosynthesis, absorption, phagocytosis, or ingestion. Color is going to be a big clue here. For example, if you see a green alga, you know that green signifies chlorophyll, so that organism is going to be a photosynthesizer. Now, not all photosynthetic pigments are green. Some photosynthetic pigments are brown, in fact, or golden. The diatoms are photosynthetic, but they don't use chlorophyll. Their pigment is brown or golden. If you'd like to earn bonus points, you can do this by looking at more organisms than 6 in lab, and of course remember that they have to be in different general groups. You can add more boxes to the assignment sheet for your bonus organisms by entering, just clicking on Tab when you position your cursor over the right box. Only one additional organism from a general group will be considered for bonus points. For example, you may have one green alga in the original 6, and then you can put another green alga that you identified in your bonus columns. Bonus points will be awarded at half the amount of the regular value of the points. Good luck with the assignment. If you have any questions, please contact me via email or talk to me after class or lab.